In this video, we will discuss about pan-cancer killing by GlyTR1 and GlyTR2. In the previous couple of videos, we have discussed about the specificity of GlyTR1 and GlyTR2. This GlyTR1 has a high specificity towards beta-1,6 GalNAc that are beta-1,6 GalNAc that are expressed on N-glycans, whereas GlyTR2 has a specificity towards TN-STN antigens or carbohydrates that has N-galactosamine or GalNAc. In the previous video, I have done a mistake by saying that GlyTR2 has a specificity towards GlcNAc, but it has a specificity towards the carbohydrates that has GalNAc. This is what I am clarifying here. So let us now see what are the results of this GlyTR1 and GlyTR2 on pan cancer or different cancers. And for the GlyTR1, we can see that their specificity compared to the control is high in almost all the different cancer cells that they have tested. And similarly for the GlyTR2 binding also, we can see the binding efficiencies for most of the cancer cells are higher. And also they have seen that some of the cancers has both the carbohydrate antigens, that is N-glycans with beta-1,6 GalNAc, and also there is GlyTR2 that has specificity towards N-galactosamine or GalNAc. And so now was verifying the binding efficiencies of GlyTR1 and GlyTR2. Now they check tumor macroarray data of the healthy conditions and also the cancerous conditions. First they check with the healthy conditions. Here the results for the GlyTR1s are shown, and we can see that for the normal kidney and also for the spleen, liver and placenta, they can see some binding of GlyTR1, indicating that beta-1,6 GalNAc are expressed on the normal livers also. And similarly they have checked it for the normal colon also. They can see some levels of bindings are there for the GlyTR1 at normal levels. When they check it for the different stages of colon adenocarcinoma, and they can see that with increase in the stage or as the cancer progresses towards metastasis, there is a high binding of GlyTR1. This is also in line with the understanding about the beta-1,6 GalNAc expressions increases with ages or as the cancer progresses. So this results seems to be in line with the previously known informations about the beta-1,6 GalNAc on the cancers. Now so here they have validated the binding efficiencies of GlyTR1 on the tumor macroarrays data. Now they wanted to check the efficiencies of this GlyTR1 and GlyTR2 transduced into the T cells, or CAR GlyTR1 and CAR GlyTR2, on the cell death of the tumors of different types. And the figures that are on the in the form of left hand side in the form of right bar, figure that is in the form of right bar are shown the cell death results of the GlyTR1, whereas figure that is shown in the blue bar shows the cell death results of GlyTR2. And we can see that in almost all the cancer cells that they have tested, they have seen the increase in the cancer cell death, both for the GlyTR1 specific CAR and GlyTR2 specific CAR. And one thing that they are able to find out that for the they have pointed out is that for the ovarian cancer, since the expression levels of beta-1,6 GalNAc are higher in vitro conditions, because in order to replicate the in vivo conditions they need to have proprietary media. So they might be over the results of the ovarian cancers or the SCOV3F plus PBMC, and the dead cells of the ovarian cancers in these conditions might be overrepresented of the actual in vivo results that people might observe when they use the GlyTR2 or GlyTR1. So this is what they have also pointed out in their research paper, so or in their publications. So however, they are able to make a point that the specificity about this GlyTR1 and GlyTR2, and also they can cause the death of the tumor cells when co-cultured with them. And those and cancer, if the cancer cells have those specificity, that is high beta 1,6 GalNAc structures in N-glycans, and also high these GalNAc structures or the TN-STN structures with GalNAc, or the TN and STN structures. So this is what they have presented in this figure. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.